Good evening to one and all present here. I hope my voice is visible, my voice is audible, and my PPTs are visible. Can anyone respond?
Yes, sir.
Okay. Okay. Then, without any delay, we will start the session. So, myself, Ajit Kumar V, a PhD research scholar from Department of Bioscience and Bioengineering from IIT Guwahati. Today, we will be seeing the Week 9 live interactive session on the course Environmental Biotechnology for the course code NOC 25 BT 80. So this session will be comprising of two main parts. First is we will be recapping the important concepts that have been covered during the week 9 lecture, followed by we will be discussing some of the sample MCQs related to week 8 and week 9. So stay tuned to answer those MCQs. It will be very useful for your final exams. So without any delay, we will start the session. And before starting the session, I will introduce myself. Myself, Ajit Kumar, a JDP program research scholar. It is a research collaboration between IIT Guwahati and IIT BHU Varanasi. So I work in a laboratory of biochemical and environmental engineering lab, where my lab mainly focuses on wastewater treatment approaches using different strategies, where my research inclines to the application of enzyme-mediated microbial degradation in the wastewater treatment, especially textile wastewater treatment. So moving on to my qualification, I have done BTech Biotechnology from Sastra University, Tamil Nadu, and MTech Industrial Biotechnology from NITK Surathkal, Karnataka. And moving on to my work experience, I worked in many pharma companies like Biocon and Enzyme Bioscience. So this is all about my personal introduction, and we will move on to the first part of today's session, which is the recap of week 9 lectures. And here we will be discussing some of the important concepts that has been covered. So, if you see in the last lecture, that is the last live interaction session, we have covered the culture independent techniques, where we have discussed two important techniques, which is amplified ribosomal DNA restriction analysis and denaturing gradient gel electrophoresis. And these two methods are mainly a culture independent technique that are used to understand the microbial community in the ecosystem. And next, we have seen some of the advanced techniques like NGS, which is next gen sequencing. And we have seen how the evolution of NGS has occurred, where we have discussed the first generation NGS, second generation, and third generation, and what is the difference between them and how it evolves during the period of course of time. And then we have seen one important concept, which is metagenomics, and how it is done and what are all its advantage, how it is used in context of microbial ecology, and we have discussed their limitations too. And there we have discussed one important concept, which is OTU, which is Operational Taxonomic Unit, and why it is important and why it is necessary to study in the environmental biotechnology course. And we have also seen several omics approaches, where we have seen meta transcriptomics, followed by meta proteomics. We have seen and meta transcriptomic analysis. So these are all the some of the important concepts that has been covered in the last live interaction session. And if you have missed out those sessions, you can always visit in the YouTube, where the recording sessions are available, or you can go to the NPTEL Environmental Biotechnology course, where a separate tab of recording sheet is there, where you can access both the PPTs as well as the YouTube lectures. So if you have missed out it, you can go and refresh it. So in today's session, we will see the most important concept, which is bioremediation. So till now, that is till week 8 of the lectures, we have seen various techniques that are used, especially in the environmental biotechnology, to understand the microbial ecology concepts. Where we have seen culture dependent techniques, where we have seen some isolation of pure culture through direct culture techniques as well as the enriched type. So, and various concepts has been used to understand or to culture these microorganisms. And we have seen some of concepts like culturable microorganism, non-culturable. In that, we have seen viable culturable microorganism and uncultured microorganism. So these are all the cultural dependent techniques and non-cultural dependent techniques. So till now, we have seen the technology or the methods to characterize them. But from now onwards, which is week 9 to 12, we will be seeing the application of these techniques, especially in the environmental biotechnology concept. There we will see all the application part, and in this week we will be discussing on the first application, which is bioremediation. So this is one of the most important application that has been widely used in order to face the challenges of the environment. So in this session, we will see the bioremediation. First, we will start to understand the bioremediation technique. So first, we have to understand what is bioremediation. The term itself self-explanatory. That is, it is two terms, which is bio plus remediation. That is, we are trying to treat the pollutant that are present in the environment using some biology source, which can be microbial or their product or their enzymes. So anything which is used to clean up the environment, we especially through biological means, we call this concept as bioremediation. So if we see the definition, so it is a managed or it is a spontaneous process. So sometimes it can be human intervention or it can be a natural process by which the biological catalyst, it can be microbes, the microbes we can say like it can be bacteria, fungi, yeast, or any other bacteria. Sometimes plants and insects are also being used for remediating the environment. So it can be any biological catalysis that acts on the pollutant. And we will be seeing what are the types of pollutant that are present in the environment. So thereby we are trying to remediate or eliminate the environmental contaminants that are present in the environment. And these contaminants can be in water, wastewater, or sludge, soil, aquifer materials, or gas stream. So this is the basic definition where we will be cleaning the pollutant that are present in the environment through various means, like it can be in water or wastewater, or it can be sludge, or it can be soil through biological catalysis. So this is a technology that used to reduce, eliminate, or contain the hazardous contaminants. So this technology we refer to as bioremediation. So, so what are all the key players that play an important role in during these reactions? Mainly the microorganism. It can be, as I said, it can be a bacteria, fungi, yeast, or any other microorganism. And to some extent, it has been extended to plants and insects. If you are using plants to remediate the environment, we call it as phytoremediation. This will also be discussed in the upcoming slides. So, what are all the key players? Microorganism, plants, or insects, which helps to transform or degrade the contaminants. And this transformation and degradation process, it can vary depending upon the environmental conditions. And what are all the environmental condition? It can be a physical chemical environment. That is physical chemical environment means it can be any factors, like it can be a temperature, pH, or oxygen level. And it also depends upon the microbial community that are present in the particular environment. And also the main factor that affects is the nature of the contaminant. That is the source of pollutant. It can be pesticides, it can be dyes, or it can be antibiotics, anything. So these are all the three important factors, and many other factors are also there. But these three play an important role in determining the transformation and degradation of the pollutant. That is, first is the physical and chemical factors, and it can be pH, temperature, or oxygen level, etc. Or it can be carbon source or nitrogen source. And second is the microbial community that are present in the environment. And third is the nature of the contaminants. Depending upon the source of the pollutant, the degradation and transformation varies. So this is the introduction to the bioremediation where we discussed what is bioremediation, how does it help to address the environmental challenges, and what are all the key players we have seen, and what are the factors that affects the bioremediation. Now we will see how the evolution of bioremediation takes place in the context of environmental biotechnology. And as we have discussed earlier, so this bioremediation is nothing but it is a pollution treatment technology which uses microbial source or their products. Product in essence, it can be some organic acids which helps to speed up the process of bioremediation, or its enzymes. The microorganisms that are producing their enzymes can help in treating this pollutant. So, which helps us to reduce, eliminate, or contain this contaminants that are present in the soil or sediments or water or air. So, normally they are trying to reduce, eliminate, or contain the pollutant. And if you see the roadmap of this bioremediation process, it has been started earlier 1900, where this bioremediation concepts has been applied only in the municipal wastewater treatment system. And gradually in 1950, it has been applied to industrial wastewater treatment process. And the scientist has come to a conclusion that and hypothesis and hypothesis on microbial infallibility. And what do you mean by this microbial infallibility hypothesis? It tells us that for every chemical that is created on the earth, there is somewhere in the nature there exists a microorganism that can break down. So what does it tell? For every chemical that we create or that we produce, there is always a microbes that lie in the earth to degrade them. That is, no matter how the tricky the pollutant are, the nature has always find its way to degrade them. So this is what the scientist tells in the microbial infallibility hypothesis. And this helps them to use this bioremediation technique in the industrial wastewater treatment. And gradually in 1970, the bioremediation has been applied to gasoline contaminated aquifer, where, for example, the gasolines contains more of hydrocarbons, hydrogen carbons contaminants. So the microorganism were able to degrade these hydrocarbons, which is petroleum hydrocarbons. And we have seen the oil spill examples earlier in the lectures. And in later 1980, what happened? Scientists were trying to understand the importance of this biogeochemical process in bioremediation. And we have already studied the biogeochemical, which is nothing but it combines the biological and geology and chemistry. That is, it helps to understand the not only the microbe between the microbe interaction, but also with the environment. So scientists were slowly trying to understand the importance of microbes and how these consortia of microbes interact among themselves and also with the environment. And this idea gave the scientists to work on real-time problems. And in later 1990, and various hybrid approaches has come to place where biological approach won't be sufficient to degrade the or mineralize the component completely. So they took help from the So they took help from the chemical process in order to aid the speed up of process of degradation. Then in early 2000, what happened? And modification of this bioremediation has been taken place, where in situ bioremediation and monitored natural attenuation techniques has been comes into play, and it was found to be cost effective and regarded as cleanup alternatives. And we will be seeing this in situ bioremediation and monitored natural attenuation in the upcoming slide. But as of now, the in situ remediation is nothing but treating the pollutant on site itself by continuously monitoring them, by enriching the microorganisms that are present in the environment. So these are all the roadmap of the bioremediation that has advanced the understanding of our microbial ecology, that paved the way for facing the environmental challenges. Then, most important is we need to understand why we need to approach for bioremediation approach. The first is, as we all know, there is a drastic increase in the pollution, not only in the air, but in also soil, surface water, and ground, which poses a major threat to the public health, not only to the public health, to the environmental health and the sustainability. That is both in the developing countries as well as the industrialized countries such as European Union states, USA, India, and China. So, due to the increase in the pollution in the last century alone, due to the human activity and due to rapid industrialization and anthropogenic activity, it has resulted in degradation of our natural resource to an approximately 39 to 50 percentage of the land resources has been drastically modified due to the human activity and due to the rapid industrialization. And also due to increase in the atmospheric CO2 concentration, which is increased by 40 percentage over the 140 years, which led to the global warming, which is a major threat to the environmental pollution as of now. And this is due to hydrocarbon combustion and deforestation. So these are all the source of pollutant causes. And what happens due to this? Due to this industrialization, rapid industrialization and human activities, the biodiversity has significantly impacted. Therefore, many natural resources has been impacting soon due to the widespread of contamination of groundwater aquifers by hazardous waste, soil, rivers, and oceans. So these are all the reasons that now scientists are approaching with the bioremediation approach. So these are all the source of the pollution and reasons for going for the bioremediation. Then, as we have know, pollutants are the major concern which has increased the pollution to the drastic level. And if you see the classes of contaminants that are affecting both soil and waters, are the first major class is that they are heavy metals. Heavy metals, in a sense, it can be any kind of metals. That is, it can be arsenic, chromium, sodium, calcium, etc. So, some metals are micronutrients, but most of the metals are toxic even in the lower concentration. And second class of contaminants is organic compounds. And organic compounds, as we already know, it is mostly composed of carbon chains. And for example, you can say the mineral oil hydrocarbons, polyaromatic hydrocarbons, benzene derivatives, and halogenated hydrocarbons. So these are all the two major contaminants that affects the soil and water. And if you see the majority of this organic compound pollutants, they are xenobiotics. And what do you mean by xenobiotics? Xenobiotics is nothing but the contaminants which are recalcitrant and persistent. And these are all foreign and man-made substance. So if you see the most of the xenobiotics are causing heavier pollutant because they are man-made pollutant. And if you see the classification and their application in agricultural field, as we all know, pesticide has been used drastically. Pesticide usage is increasing for better crop yield, right? So, but these pesticides, if they are present at very higher amount, it causes pollutant. And if you see some of the examples of these pesticides, it is dichloro, diphenyl, trichloroethane, which is DDT, atrazine, and pentachlorophenols. And if you see in industrial fields, the solvents such as dichloroethane or dielectric fluids such as polychlorinated biphenyls, these are all regarded as the organic pollutant which are considered to be xenobiotic. And if you see in terms of military use, explosives like 2,4,6 trinitrotoluene, these are all the chemical compounds that are used for explosive. These are also a in pollutant that are used in the military use. And if it is a very small amount, it causes drastic pollution to the environment. So these are only some of the xenobiotic contaminants that has been used in various applications. And among the pollutant that has been seen, as of now, some pollutants are emerging contaminants. And what is emerging contaminants? These are all substances that are present in the environment for a long period of time, but their presence and their negative effect have only recently been recognized. That is, this substance has been present in the environment for a long period of time, but it has been unnoticed for a long time. So now it has been come into a concern because their presence are very minute in the environment, but their effect is now alarming and causes pollution to the environment drastically. So if you see these emerging contaminants, these are contaminants that are not regulated by the government bodies. They are still in the discussion and we need a huge amount of data and research in order to form a regulation for this pollutant. And if you see some of the examples of this emerging pollutant in various fields, for example, if you see in the human and veterinary pharmaceuticals, we all know that there is a huge usage of antibiotics, right? Antibiotics. These antibiotics is also regarded as one of the emerging contaminants. And this anti-parasitic agents and ionophores. These are all some of the substance that has been widely used in the human and veterinary pharmaceuticals. And if you see in the stimulants, stimulates and a drug anti-inflammatory, anti-diabetic, antihypertensive, anti-cancer drugs, anticoagulants, and analgesics. These are also regarded as the contaminants, but their presence is very minute quantity in the environment. And these are all the contaminants which are majorly present in the pharmaceutical wastewater. And hormones, including natural and synthetic estrogens and androgens, these are also present in the pharmaceutical and hospital wastewater. And if you see the industrial and household wastewater, insecticide, plasticides, detergents that we use normally in the household purpose, flame retardant, polycyclic aromatic hydrocarbons, antioxidant, fumigate, and preservatives. These are all some of the industrial as well as the household wastewater products that now emerge as the emerging pollutant. And if you see in the personal care product, this insect repellent, polycyclic musk, sunscreen agents, fragrance, and antiseptics, these are also now emerging as the personal care product. So from these slides, you need to understand that these are all like day-to-day products, but we are not aware that it is also one of the emerging contaminants because their presence is very small and we are unnoticed of these pollutant. And now the government bodies like United Nations Environmental Pollution Agencies and several pollutant agencies are taking this as one of the major concern. And now this was regarded as the emerging contaminants and lot of researchers are working on these wastewater wastewaters and finding a viable solution to handle this emerging contaminants. So this is also one of the major threat to the environment. And in order to deal these organic pollutant as well as the emerging contaminant, we need a viable solution in order to tackle them, right? So what is a viable solution that needs to be cost effective and it needs to be eco-friendly as well. So we don't have none other option that is at last we end up with microbes. That is microbes, particularly bacteria, the most effective candidates for handling these kind of bioremediation technology. What happens here? These are all the pollutant. It can be alkane, alkene, alkyne, and aromatic hydrocarbons. These are all regarded as the organic contaminants. These organic contaminants will be utilized by the microbes, which is regarded for them as the source of carbon as well as the energy source. Once these microbes consumes this carbon or energy source, eventually they will be converting them or degrading them into carbon dioxide and water. Sometimes intermediate metabolites can also be formed either in the aerobic or anaerobic condition. So what happens? The microbes will try to degrade these toxic contaminant into simple compounds like CO2 and H2O. If it is converting them to CO2 or H2O, we call this process as biomineralization, either in the presence of oxygen or in the absence of oxygen. So this is how the microbes helps in degrading this pollutant. And in the later slides, we will be discussing the mechanism, how this microorganism acts on this pollutant we will be seeing. So why these microbes needs to be used? The first and foremost reason is that this is the major entity that causes large scale transformation in the biosphere. And if you see the biosphere, they are the invisible ruler of planet. So, and also, these are all the first life form that evolved in the Earth, that is 3.8 billion years ago. And they are ubiquitous, that is, the microbes are literally everywhere. They are present everywhere. Think of like it is an nature Wi-Fi. That is, it will be present everywhere, even if we can't see them. And third most important is they are most abundantly available and metabolically most versatile. That means they can eat anything, they can consume any kind of pollutant, such as it can consume the carbon source like sugar as well as the pollutant like emerging pollutant too. So it is metabolically most versatile and it is as the highest genomic diversity and the microbes make up the two-thirds of the entire domain of life. And also the microbes, especially the prokaryotes, that is bacteria, they have the small genome size. So it simplified the process and they are very compact and they replicate very quickly. And they have short replication time as well. That is, there are some bacteria like E. coli, they have a doubling time of 20 minutes and most often they rapidly adapt and evolve themselves to the new environmental condition. So these are all the some of the advantage for going for the microbes, especially prokaryotic microbes for bioremediation of pollutant. And if you see why bioremediation needs to be applied for the treating the pollutant, the first is their versatile of the microbial process to withdraw or to withstand the most adverse condition. And most often, microorganism they tolerate as well as they detoxify the multi-component. Some bacteria can degrade multi-contaminants, that is one class of pollutants simultaneously. And if you see, most often there won't be no secondary waste generation because, for example, if microorganism A degrades a pollutant which converts into intermediate components, These intermediate components will be consumed by the other group of microorganisms in order to mineralize them completely. So most often what happens here is there will be no secondary waste generation happens, but in rare cases some waste will be generated, but eventually it will be treated with other group of microorganisms that will be mineralizing this secondary metabolites or intermediates. And this, especially this bioremediation, it is useful for high volume dilute waste where the trace contaminant limit the acceptability of the final waste. And as they are more economical, that is, they are most cost effective when compared to the other physical and chemical process. And this is mainly applicable for non or slowly degradable organic and inorganic contaminants. And this is why we need to apply the bioremediation technique for handling the organic pollutant. And if you see the advantage of bioremediation, the first and foremost advantage that we have seen is it is more efficient and economic. As we have discussed earlier, during the oil spill that occurs in the ocean, some microorganisms, what happened is they clean up the pollutant efficiently, and it was found to be low cost when compared to the other physical and chemical method. And it most often we have seen that it is no secondary waste will be generated. What happens? The microbes try to consume the pollutant and convert them into harmless product like CO2, water, or non-toxic intermediate product. And most often it deals with the multiple component in the hospitality condition. That is, they have the supreme adaptability behavior. That is, it can clean wide range of pollutant across the environment, and it take care of even slow degrading and inorganic contaminant. That is, some pollutant may be recalcitrant. That is, they are often difficult to degrade. So what happens here? The microorganism will tend to produce unique enzymes that can act slowly onto the pollutant and degrade them efficiently. So these are all the some of the advantages of the bioremediation technique, which is they are efficient and economic. There will be no secondary waste, and also it not only deals with the single contaminant, sometimes it can deal with multiple contaminants in the adverse condition. And bioremediation can be applied to even slow degrading and inorganic contaminants. So these are all the advantage of bioremediation. And if you see the application of this bioremediation, it not only applies to the pollution, it also extended to the other fields also. And we have seen first is removing the pollutant. Other than this, it also helps in valorization of waste chemicals. That means that is the waste can be turned into something useful. That is, waste is converting into wealth. For example, we can convert the cooking oil into biodiesel. So we are trying to convert the waste into wealth. And also it ensures the circular economy. And the term circular economy denotes that nothing is wasted. So it is mostly recycled back without any wastage. So these are all the some of the applications that bioremediation helps in tackling the environment. And if you see the environmental fate of environmental contaminants, there are two major factors that governs the environmental contaminants. One is abiotic factors, and as we have already seen the abiotic factors in the early lectures, and can you name any abiotic factors? Abiotic factors can be abiotic factors can be it can be either temperature, pH, or oxygen level, anything. Whereas biotic factor represents the microorganism. It can be prokaryotic, eukaryotes, it can be yeast, fungi, anything. So any living matter is regarded as biotic factor, Any non-living matter, which is the environmental condition most often, it is regarded as the abiotic factor. And these two factors determines the fate of environmental contaminants. And if you see the microorganism, when it acts on any pollutant, it can be organic pollutant or metals or radionucleotides, the bioremediation, the progress of the bioremediation mainly depend upon these three factors. One is their chemical property, how the chemical structure, the charge, and many factors like the pKa value, the pH of the particular pollutant in the environment. It all determine the degradation or progress of the bioremediation process, and also their environmental mobility or persistence. So it also depends upon how they react or present in the environment. Some pollutant can be persist for a long period of time. Then concern must be raised in order to isolate a particular microorganism that can degrade this pollutant in a very short period of time. And their toxicity, because some, especially in the metals, in case of metals, uh, metals, even in the millimolar concentration, they are toxic to the microorganism as well as to the environment. In such case, we need to find a strategy to eliminate these metals, but metals can't be eliminated as such. So that what happens here is the scientists were trying to transform these metals to the very less and non-toxic form. Very less and non-toxic form. So what happens here? So for example, we can take any one of the metals, which is chromium 6 plus. Its oxidation state is 6 plus. So this chromium 6 plus is very toxic even in the very low amount, that is in millimolar range. So what happens? The scientists were trying to decrease their oxidation state to 3 plus so that it might be less toxic and acceptable to the environment. So these are all the three important factors that determines the progress of the bioremediation. The first property is that their chemical property. The next property is their persistence of the particular pollutant, and third is the toxicity of the contaminant. And if you see how these microbes interact with the organic or metallic contaminants, so these are all some of the strategies that microorganisms use in order to clean up the pollutant. So we will see the strategy one by one. The first strategy is their biodegradation. So the term itself telling us that it is degrading the contaminant. That is, it is breaking down the organic compounds into simple and harmless product like CO2, H2O, etc. For example, we can take the same example of oil spills and what happens when oil spill in the ocean. They use a specific type of microorganism. It can be, for example, it can be a Deinococcus microorganism. These microorganism, what happens here is they take up these hydrocarbons because petroleum contains high amount of petroleum hydrocarbon chains. So these microorganisms will eat up these hydrocarbons and they convert to carbon dioxide, water, and biomass. And this strategy we call it as biodegradation, where the complex contaminants will be converting into simpler products. And this is one way where the microbes interact with the organic compounds. Whereas if we take the biotransformation, the second case, what happens? This is mostly seen in the metals, that is heavy metals. And we all know that heavy metals can't be degraded completely, but it can be, we can change its form. That is, if you see in the biotransformation mechanism, where metals either oxidized or reduced. For example, we will see the take the chromium as an example. The chromium, the 6 plus oxidation state, it is very harmful. But after biotransformation, their oxidation state is get reduced to 3 plus, which is less toxic, but in the safer form. But in the safer form. But in the safer form. So here we are trying to transform the metals, not by eliminating them. This is one of the strategy that uses microorganism to interact with the metals. Whereas in the third strategy, it can be bioaccumulative. What does it mean? It will try to trap the metals inside them. They don't degrade, they don't transform, they don't do any such of thing. They just accumulate these metals into their cells. Or it can undergo biosorption, where what happens, these metals will be adhering to their surface. It is like adsorption mechanism, where the metals ions, they don't degrade, they just accumulate and adsorb onto the surface. Whereas in bioprecipitation, they tend to precipitate these metals so that separation can be easier. So from this slide, we could understand there are various interaction of microbes with the organic pollutant as well as the metals. And first we have seen the biodegradation approach, where complete breakdown of the metals occurs, whereas in the second strategy, where biotransformation is happening, where scientists were able to trying to transform the metals into another state which is acceptable and in the safe limit. Whereas in the third stage, either they try to accumulate the metals inside, or they try to absorb these metals onto their surface, or they try to precipitate their metals so that the separation can be easier. So these are all some of the interaction technique that are seen in the environmental pollutant. And if you see, as we already know, the microbes, they often see these pollutant as the valuable resource. And these are all some of the factors that you need to know before going to the next concept. First is, we need to understand their metabolic strategy. That is, every microorganism has their own tricks to have their food as the pollutant and the energy resource as the pollutant. So every microorganism, they have a different metabolic strategy to handle this pollutant as the resource. And in addition to that, you also need the organic compound, that is organic pollutant, as their food, that is their packet of energy as well, because these helps to break down these organic compounds for building their cells as well as for energy to grow. And third is the heavy metals, and we already know heavy metal often it is regarded as electron donor. Sometimes it can act as an electron acceptor or the micronutrient, because if it is present in a very high amount, it again became toxic to the microorganism. And the other inorganic sources like nitrogen, phosphorus, sulfur, these can also act as an either nutrient or electron donor or electron acceptor. Because, for example, the bacteria can use the nitrogen compound not just for their survival, but also they try to remove the excess nitrogen if it is present excess. And third is their detoxification strategy. What happens? Most often they try to neutralize the toxicity of the pollutant by either trapping them or changing its form, as we have seen in the bioaccumulation or bioabsorption and also in the biotransformation. So every microorganism has their own detoxification strategy, and also the co-metabolism. That is, the microbe degrade the degrade this pollutant while eating some other organic compound. For example, bacteria can digest the methane, but simultaneously it can also have the capacity to degrade pollutant or pesticides in the soil. So these are all the some of the co-metabolism strategy that microorganism play and uses pollutant as the valuable resource. So these are all the different strategy that microorganism undergo in order to progress the bioremediation. And if you see the advantage of this bioremediation, the first and foremost advantage is that cleanup can be done in situ, which eliminates the hazardous waste transportation and liability cost. Viability cost. That is, what happens here? The cleanup, the treatment will be continuing in the on-site itself where the pollutant is present. So there is no need to dig up the contaminated soil or pump out the water for treatment. So the treatment can be done in the location of the contaminants itself. So this is one of the advantage. And second advantage, the organic hazardous waste can be completely destroyed and it can be converted into water, carbon dioxide, or many other mineral source rather than we are just transforming from one phase to the another phase as we see in the chemical or physical treatment. So what happens here? We could get a complete destruction of organic hazardous waste where these organic waste will be transforming into the harmless products like CO2, water, and mineral salts. And third is they rely on natural biotransformation process that can be faster and cheaper. That is, these process normally they are faster and cheaper through natural process. That is, they break down the contaminants using the metabolic proteins or their enzymes so that fewer chemicals or less energy and low cost will be used when compared to the other techniques. And also minimum land and environmental disturbance will be happening. That is, traditional methods like excavation and pumping, it causes disturbance to the environmental as well as it is very cost expensive treatment strategy. Where with the advent of in situ techniques, the progress of the bioremediation happens in the on-site itself so that the excavation and the pumping costs get reduced drastically. And also it can be applied to hard to withdraw hydrophobic pollutant. That is, this bioremediation technique are also effective against the hydrophobic pollutant. That is, the pollutant stick tightly to the soil or sediment and difficult to remove. In that case, also microbes can sneak in and break down this pollutant effectively and convert them into simple and non-toxic form. And sixth point is that they are environmentally sound with public acceptance. That is, they are more eco-friendly and now it is publicly accepted for going for bioremediation for cleaning up the pollution. And also doesn't dewater the aquifer due to pumping. That is, it doesn't require dewatering of aquifers. That is, pumping lowers the water level, disturbs the ecosystem, increases the cost. But these microorganisms doesn't involve any dewatering of aquifers. And last and most important advantage is that they can be used in conjunction with the other treatment technology. As we have discussed earlier, along with the biological treatment, some simple physical and chemical treatment can also be integrated in order to speed up the progress of the bioremediation so that degradation or the progress of bioremediation can be speeded up. So these are all the advantages of the bioremediation and as well as some of the disadvantages also there with respect to the bioremediation, that is certain pollutant, for example, the heavy metals, they are not completely eliminated by this biological process. That is, they are not completely destroyed by the microbes. What happens, at least what we can do is they can transform there or change to less toxic or less mobile form. So this is one of the major concern when we are dealing with the heavy metals. And as we have seen in the mechanism part, we can transform these heavy metals to the less toxic. As we have discussed, the chromium 6 plus is transforming into chromium 3 plus. But this 3 plus will exist in the environment, but it is regarded as less toxic when compared to the original parent compound. And also, it may require extensive monitoring depending upon the process that we choose. That is, it requires extensive monitoring, continuous monitoring of pH, water, temperature, oxygen level needs to be monitored for effective degradation. And the requirement for success and removal efficiency may vary from one site to the another site. So this is site-specific success too, because the efficiency of the bioremediation can vary from one place to the another place, because here various factors comes into play. The first is it can be the soil type that we are handling, because the soil type, the morphology, the soil content may vary from place to place, and also the temperature and pH of the atmosphere determines the progress of the bioremediation, and also the nature of the contaminant that are present is also determines the success of bioremediation. So it often vary from one side to the another side. And also some contaminants can be present at higher concentration that can inhibit the microorganism. This can also happen because sometimes in particular environment there could be high presence of metals. So at very high level, what happens due to the high concentration of these metals, it can kill or inhibit the microorganism of our interest, so that the degradation efficiency will be lowered drastically. And also it can be scientifically intensive technique. That is, you need advanced knowledge of microbial ecology, biochemistry, environmental biology. So this indicates that you need skilled labor or scientists and infrastructure in order to handle this bioremediation technique. But it doesn't apply to all the techniques, but it mostly applies to the ex situ as well as the natural attenuated in situ. And there is a risk for the accumulation of toxic biodegradation product. That is, there may be due to, as we have seen in the earlier, bioaccumulation is also one of the strategy where microorganisms tend to accumulate this metals inside the cell. What happens here? Still, the metal is present inside them. So this should be managed carefully, and this should be released into the environment unless we perform the phytotoxicity analysis before discharging them into the environment. So these are all the some of the disadvantage of bioremediation, but ultimately this offers, and this is only viable technique because of their economy purpose and also their eco-friendly approach. So if you see the types of the bioremediation and approaches of this bioremediation, types and approaches of the bioremediation, the first technique is that we have to understand what is the difference between the in situ technique and ex situ technique. So what is in situ? The term itself denoting it is in. That is, the remediation will be taking on the on-site. That is at the location of the treatment. So the contaminants remains in the place during the bioremediation. We call this as the in situ process. Whereas ex situ means we will be taking out or excavating this pollutant and will be transporting to the another place for treatment of pollution. So we call this step as the ex situ process. So first you have to understand the difference between the in situ remediation and ex situ remediation. In situ remediation denotes the treatment will be carrying out at the location of the treatment itself. Whereas in the ex situ, what happens? We will be removing the contaminants through excavation and we'll be treating them in the separate place. So we'll be collecting and treatment will be in the separate place. We call this process as ex situ process. And the other approach is that intrinsic and engineered process. And what do you mean by intrinsic? So here we will rely on intrinsically occurring rate of supply of substrates as well as the nutrients as well as the intrinsic population density of active microorganism. That is, we will be using the natural nutrients and the energy available for the naturally available microorganism to transform the contaminants to the harmless product. This approach is often slow, but it works if the correct microbial population as well as environmental conditions are present in the particular environment. And we call this kind of bioremediation as intrinsic bioremediation. Whereas in the engineered bioremediation, we will be using advanced genetic engineering tools to increase the progress of the bioremediation. What happens here? We will be using the principles of genetic engineering and advanced tools to speed up the process of progress of the cleanup of contaminants. So these are all the four terminology that you need to understand before moving on to different techniques. That is, in situ treatment will be performing on site itself, whereas ex situ means we will be taking out the contaminant and will be progressing the treatment system. And intrinsic is we will be providing all the nutrients as well as the condition, whereas engineered means we will be using the engineered species, using the genetic engineering tools to speed up the process of the progress of bioremediation. And if you see the strategies, first is in situ. As we already know, the contaminants will be remain in the place during the bioremediation, whereas in the ex situ, we will be first excavate and transport them to the another place where the process will be taking place. Whereas in engineered, we will be using some engineered tools to increase the input rate of the degradation mechanism. Whereas in the intrinsic, we will be depending on the naturally available microorganism. We will be providing the nutrients and environmental condition to increase the success of the bioremediation. And these are all the techniques and the approaches then one generally follows. And if you see the classification, there is another important classification which is biostimulation and bioaugmentation, and we will be seeing in the coming slide. But this is the overall classification of bioremediation, where in situ again it has been subclassified into intrinsic and engineered, where in engineered we have biostimulation, bioaugmentation and biobarrier, whereas in ex situ we have composting, bioreactor and bioland forming. And we will be seeing these differences in the upcoming slides. And first concept is that biostimulation, bioaugmentation. So what does it mean? Here, it is a process of adding the culturable microbial population which have the ability to degrade the specific contaminant. That what happens? So it is like an in situ technique where you will be adding the appropriate microorganism to the environment to proceed for the bioremediation approach. So what happens here? Here you will be adding the natural strain or it can be genetically engineered strain with unique metabolic profiles that will be added to the contaminant site to supplement the indigenous microflora and speed up the biodegradation. And what are all the common applications? It can be used for chlorinated contaminants, petroleum hydrocarbons, etc. And the microorganisms, usually they used to isolate from the contaminant itself or from the historical site, or they will use the genetically modified strains to support the remediation process of contaminant site. So in order to give a glimpse, bioaugmentation is nothing but you will be adding the culture to the contaminant site and you will be providing the nutrients as well in order to speed up the progress of bioremediation. For example, we will take again the oil spill. What is the resource here? Here the resource is hydrocarbons. So the microorganism that we are using, it can use the hydrocarbons, petroleum hydrocarbons as the food, petroleum hydrocarbons as their food, and it helps us to remove these pollutant effectively. So this is what we call it as bioaugmentation. Then what is biostimulation? The term itself giving us the idea that it is stimulation. So the microorganism will already be present in the environment. So what we need to give? We need to give only the nutrients. to support their growth and to give energy for degrading the pollutant. So here the microbes cannot use pollutant as only the source of energy. So what happens here? We will be adding the supplemented nutrients. So biostimulation is nothing but it is a process of environmental modification. So we are trying to modify the environment by adding the limiting nutrients, electron acceptors like it can be nitrogen, phosphorus, mineral salts. Sometimes it can be oxygen, carbon in order to stimulate the existing microbial population which are involved in the bioremediation. So this is the most common technique that we use against the petroleum pollutant in soil. So if you see in the right hand side, so this is the contaminant environment. So if I give a one of the contaminant environment and if I say you have to use bioaugmentation approach means what you will do is apart from the microorganisms that are present in the environment, you will be using a particular microorganism that has the capacity to degrade these kind of pollutant. So you will introduce the strains in order to degrade the pollutant as fast as possible. So this concept we call it as bioaugmentation. Whereas in biostimulation, what happens? So there will be already an indigenous microorganism that are present in the environment. What do you mean by indigenous microorganism? The microorganism that are already present in the site of the pollutant. So what we will do is we will provide appropriate nutrients or environmental condition to promote their growth as well as to speed up the process. So by these two approach, what we ultimately get, we will be ultimately removing these contaminants. So this is the two important concept or approach that scientists normally use to face the environmental challenges. One is bioaugmentation and one is biostimulation. And other approach is that as we have seen in the earlier, not only microbes, sometimes they use plants also in order to clean up the environment, right? So in this case, if we use the plant or their products for the decontamination or stabilizing the contaminants and the metals from soil, we call this concept as phytoremediation. That is, remediation means cleaning the environment from the pollutant by using the plant or their product. So then what is the difference between bioremediation? Bioremediation is cleaning the pollutant using the microorganism. So if you use microorganism, it is bioremediation, and if you are using especially plants, they are called phytoremediation. So there are lot variety of plants which has the ability to take up these heavy metals through their root via soil, and they used to accumulate them in the stem, shoot, and leaves. So we call this concept as phytoaccumulation. So these plants, what they possess a particular gene that regulates the amount of metals that taken up from the soil through roots, and they deposit to the other location like stem, shoot, and leaves. So, so if you are using the plant as one of the source to clean up the pollutant, we call it as phytoremediation. And depending upon the process, applicability, and type of contaminant, we can use different types of pollutant or different types of plants. For example, in the right hand side, you can see in phytoremediation, there are lot new concepts where first, if you take the phyto extraction, where what happens here is the metal used to accumulate and the scientists were able to extract these metals from them. Whereas in phytostimulation, we will be degrading the organic pollutant by the plant root exudates, that is through rhizobacteria. And phytodegradation means directly using the plant to degrade the organic pollutant. What happens here is the plant will uptake the pollutant, and in turn the plant will produce some metabolites or products that helps us to degrade these organic pollutants. And another concept is phytovolatilization, where plants tends to convert these metals or metalloids to less toxic form. This is similar to the concept of biotransformation, where microorganisms try to transform the heavy metals, that is Cr6 to Cr3. So likewise, the plant converts the metals or metalloids to less toxic or volatile form. So these are all the different strategies scientists used while applying the phytoremediation technique. So till now we have seen biostimulation, bioaugmentation and phytoremediation technique. And if you see the intrinsic way of bioremediation, it is nothing but what happens, it is intrinsic bioremediation. That is, it occurs in situ. And also they rely on naturally occurring biological process that are carried out by the indigenous microorganism. So this is a component of natural attenuation which includes the both physical and chemical process. And here main motto is to clean up the activity that rely on natural attenuation to reduce the contaminant level, and continuous monitoring will be happening to determine the effect of remediation. And we call this term as monitored natural attenuation. What does it mean? So scientists will be monitoring continuously their pH level, temperature, or oxygen level in order to measure the progress of the bioremediation in the real time. I hope now you understand the term intrinsic bioremediation. That is, they helps us to convert the environmental pollutant to non-toxic form through inherent indigenous microorganism. This process is most effective not only in the soil but also to the water, as these biomes will have higher chance of being contaminated by contaminants and the toxins. And this process usually carry out in the underground places like underground petroleum tanks. We have seen the oil spill cases, and there is escalating attention on the intrinsic bioremediation for control of all these contaminants at the waste site. Most importantly, the natural ability of this microorganism to degrade the contaminants, they are continuously monitored and examined, and they are tested at the laboratory level before releasing into the environment. And there are several conditions of the site that promotes the intrinsic remediation. As I said, conditions like their pH level, temperature, their oxygen content, their soil content, these are all determines the progress of the intrinsic bioremediation approach. And if you see the extrinsic bioremediation, it is quite opposite to the intrinsic, where what happens, we will be first removing the contaminants from the environment. Then we will be treating these wastewater by adding appropriate microbes or nutrients to speed up the degradation process. And if you see the ex situ bioremediation, it often helps to eliminate the waste materials and their collection from the contaminant site to assist the microbial degradation. And these technique always has a demerits and limitation, as we understand that uh, these contaminants needs to be taken out from the site. So this is going to be an expensive process depending upon the amount of volume of samples that we are dealing, because it needs to undergo various steps like excavation, screening, and fractionation, mixing, homogenizing, and final disposal. And more often, these contaminated material can be either liquid or it can be soil sample. So on the basis of the phases of materials under treatment, these ex situ bioremediation can be classified into solid phase system and slurry phase system. If it is a solid phase, we can go for land treatment and soil piles, that is land composting. Or if your contaminant is like slurry phase system, you can go for bioreactor in order to treat this contaminants. So these are all the two important classification in the extrinsic bioremediation, where you will be seeing land composting and bioreactor play an important role. And if you see the challenges of this bioremediation and scope of microbial ecology, the main challenges is that what works here may not work there. As we have seen the disadvantage of bioremediation, so the bioremediation progress or success changes from one location to the other location. That is a, a single strategy. There is no single strategy that will apply to all the location. Those strategies may succeed in one place, may fail in the other places. And also the lab versus field. That is in lab. For example, in the lab, everything is controlled. That is, you will have a controlled temperature, pH, oxygen, or nutrient. So the microbes will thrive and happily grow and degrade the contaminants. But when we are moving to the real-time condition where due to the complex nature and the environmental contaminants, things gets complicated. So we could not get an accurate results as we seen in the lab studies. So the main challenge is it can success can change from one location to the other location, and also there is a drastic changes in the lab versus field studies. And if you see the major limiting factor is that the microbial growth and activity, this aspect we don't fully understand how these microbes grow and how they behave in the contaminant site. This needs to be understood completely before going for taking a particular mode of bioremediation. It can be intrinsic, it can be in situ, it can be ex situ, it can be engineered approach, and it can be biostimulation, augmentation, like that. So unless until you understand the microorganism and their environment contaminants, we cannot fix a particular type of method. So from this you could understand it is very challenging and remind us that the nature is very complex. To unlock its potential, we have to study and get deeper insights on the microbial ecology. Then only we can understand these tiny organisms and how we can use them to solve the environmental pollution. So these are all the challenges that one need to take care while approaching for correct bioremediation technique. So this is all for the today's lecture. And in today's lecture, we have discussed the main topic of bioremediation, and we have seen what are all the contaminants, classification of contaminants. We have seen, and we have seen the emerging pollutant concept as well. And also we have seen why microorganism needs to be used for bioremediation, and we have discussed the advantage of bioremediation and what is the fate of environmental contaminant and what are all the microbial interaction not only with the organic contaminants but also with the metallic contaminants. And also we have seen advantage and disadvantage of bioremediation, and we have seen various types and approaches where we have discussed in situ and ex situ process, and also we have discussed intrinsic and engineered process and approaches like biostimulation, bioaugmentation, as well as phytoremediation technique. And last, we have ended with what are the challenges of the bioremediation and how it is important to understand the microbial ecology. So these are all the highlights of the today's lecture. So now we will move on to the MCQs, the sample MCQs. So for this, uh, I will be showing the question first, and you can type your answer in the chat box, or you can unmute your mic and you can tell the answer also. So if you are not comfortable, you can post your answers in the chat box so that I will view and we will discuss on it. So till now, if you have any queries, do you have any queries? Okay, Shuni, you have any queries till now? Can we move on to MCQs? Can we move on to MCQs? Okay. First question is, which of the following statement about DNA sequencing method is false? Carefully read the question. Which among the following statement about the DNA sequencing is false? Option A, Sanger sequence is still a preferred strategy to sequence short DNA fragments at small scale. Option B, second generation sequencing offers high throughput short read sequencing. Option C, third generation sequencing offers high throughput long read sequencing. Option D, third generation sequencing has higher accuracy than the second generation sequencing. Option E, Human Genome Project used first generation sequencing. So, among these statements about the DNA sequencing method, which is false, you can start to post your answers in the chat box, or you can unmute your mic and you can answer the question. And your answers are okay. Option D. Any other answer? You can view the question, you can answer it. Which among the following statement about DNA sequencing method is false? And your answers are any other answer? Option D. Okay, Ashwini and Teddy. Okay, now we will see the answer. Yes, it is correct. Option D. Now we will see why option D is wrong. Because third generation sequencing has lower accuracy than the second generation sequencing because they have high error rate. That's why the option D is false because third generation has lower accuracy than the second generation. And now we will see why other options are true. So if we see the Sanger sequencing, it is highly accurate, especially for short fragment. And till now, it remains a golden standard technique for DNA sequencing. That's why option A is correct. If you see option B, the second generation allows for massive parallel sequence of short fragment. So that's why the second generation offers high throughput, especially for short read sequence. And option C, the third generation enables the sequencing of significantly longer DNA fragment. That's why it is applicable for high throughput long read sequence. And option E, the first human genome project, it was solved using the Sanger sequencing. And if you have gone through the lectures, that is evolution of next gen sequencing, the Sanger sequencing comes under the first generation sequencing. That's why option E is true. So among this statement, option D, which is third generation sequencing has higher accuracy, is the false statement because it has lower accuracy. So you both told the correct answer. Now we will move on to the next question. The next question is, which of the following cannot be deducted using the clone library preparation? So you can carefully read the question, which is which of the following cannot be deducted using the clone library preparation? Option A, pathogen. Option B, rare biosphere. Option C, antibiotic resistant microorganism. Option D, eukaryotes. Option E, sulfate reducer. So from this statement, which of the following cannot be deducted using the clone library preparation? You can post your answers. Your answers are, you can take your time. Which of the following cannot be deducted using clone library preparation? Option D, okay, Ashwini, and option B, okay. Now we will see the answer. Correct option is option B, rare biosphere. Which of the rare biosphere cannot be deducted using the clone library preparation? Why it is so? Because generally what happens, clone library preparation, it can be done for any group of microorganisms. If they have the, if one designs the primer accurate. But however, if the microorganisms that are present in the small number are overshadowed by the more abundant microorganism, and thus the rare biosphere became very difficult to detect from the clone library based analysis. That is, if you have a large microorganism, what happens usually, the very less abundant microorganism will be overshadowed by the large abundant microorganism. That's why it is very difficult to detect these microorganisms from the clone library based analysis. And now we will see why other options can be deducted using the clone library preparation. The first is the pathogen. It is readily deducted using the clone library preparation because it aims to identify the specific microorganisms that are present in the sample. That is why it is very easily detected using clone library preparation. Whereas the option C, antibiotic resistant microorganism, what happens? The microorganism that are resistant to the antibiotics very easily. So this is also can be easily identified if scientists design the primer easily. And third one, eukaryotic clone library can be used to study this eukaryotic organism as it focuses on specific genes or expressed transcript. So this can be easily detected using the clone library preparation. And sulfate reducers, what happens? These are the specific group of microorganisms that can be identified and characterized. So option A, C, D, E, it can be easily identified by cloning them in the appropriate plasmid. Whereas rare biosphere, due to the overshadowing of the most abundant microorganism, it cannot be detected as easy as the other options. That is why option B is correct. So now we will move on to the next question. Next question is OTU, or best described as. And I hope you know the expansion of OTU, which is Operational Taxonomic Unit. So it is best described as option A, group of similar species. Option B, group of similar strains. Option C, group of identical cells. Option D, group of similar DNA sequence. Option E, group of identical DNA sequence. So among these statements, which is best suited for describing the operational taxonomic unit? Now you can post your answers. So OTU, or best described as, and your answers are, option D and option A. Okay. Option D, group of similar DNA sequence. Option A, group of similar species. Okay. Okay, now we will see the solution. The correct answer is option D, group of similar DNA sequence. Why? Before that, we have to understand the term OTU, which is Operational Taxonomic Unit, where we are more concerned on DNA sequence. And in the early lecture also, we have described many OTUs. So they segregate the DNA sequence based on their identity. That is, they are similar to certain degree. Conventionally, a threshold of 99% identity used because earlier 97 is used. Depends upon the samples and the sequence that you need to use. It varies, but traditionally above 97 is 97% is preferred. So OTUs are nothing but it is a similar DNA sequence, not the identical DNA sequence. Now we will see why other options are wrong. So if you see the option A, while the OTU represent the group of similar sequence, but they are defined at the DNA level sequences, not at the species level. That is why it is more related to the similar DNA sequence rather than the similar species. And if you see the option B, strains are nothing but it is a genetic variant within the species, right? So if option A is wrong, then obviously option B is also wrong. So it is not defined by strain similarity. And option C, based on the DNA sequence, we call it as OTU, not cell identity. That's why option C is also false. And if you see the option E, when do we call an identical sequence? If both sequences are exactly matches to one another. So then that would be same sequence, right? Not similar sequence. That then it doesn't group into multiple OTUs. That's why option D, group of similar DNA sequence, best describes the OTU, which is operational taxonomic unit. I hope now you understand the difference of OTU and the concept of OTU. Now we will move on to the next question, which is the lowest taxonomic level till which we can usually reliably identify a microorganism using 16S rRNA amplicon sequencing is, I will repeat the question, the lowest taxonomic level till which we can usually reliably identify a microorganism using 16S rRNA amplicon sequencing is option A variant, option B strain, option C species, option D genus, and option E family. So among these options, which is the lowest taxonomic level, which we can usually identify microorganism using 16S or RNA amplicon sequencing. Now you can post your answers. Option D and option C, option C and option D. Okay. So let's see the answer. The correct answer is option D, genus. We can identify till the genus level accurately. Why? Because this is one of the limitation of 16S RNA as a taxonomic marker. Usually what happens, we can identify only to the genus level. Why not species level and why not the other one? We will see now. First option, which is variant. Variant is nothing but the species, two different species with slightly variant in the DNA sequence. So with 16S rRNA amplicon sequencing, it doesn't provide resolution to the level of distinguishing specific variant among the strains. So that's why 16S cannot tell and identify the microorganism. And with this strain, it also lacked the resolution of identification at the strain level. And if you see the option C, species level identification, especially with 16S RNA, it is way more challenging, especially for closely related species, due to high 16S rRNA gene sequence similarity. That is why species level identification is very difficult. And next, if species level identification is very difficult, next taxonomic level is that genus. That is why till genus we can conveniently identify a microorganism using the 16S rRNA sequencing approach. So family, it provides the resolution beyond the family level, often up to the genus level. That's why the lowest taxonomic level, if you see in the uh, hierarchy, genus up to genus level, we can comfortably identify the 16S or RNA amplicon sequencing. We will move on to the next question. Next question is, assume that ABC1 gene is a conserved gene responsible for antibiotic resistance in bacteria. So now we are doing an experiment where we are submitting the sequence for NGS based amplicon sequencing, where both 16S rRNA gene and APC gene was targeted in a separate run. So when the data is compared and interpreted, which one of the following is not a likely outcome? First, you understand the question, read the question twice or thrice, and then answer it. So among this statement, which is not likely an outcome. Option A, 16S or RNA based analysis will give more OTU than ABC1 based analysis. Option B, 16S or RNA based analysis will reveal taxonomic composition of microbial community. Option C, ABC1 based analysis will reveal abundance of antibiotic resistant gene carrier. Option D, this study cannot determine which microorganisms are carrying the ABC1 gene. And option E, this study can determine if ABC1 gene is horizontally transferred. So among this statement, which is not likely to happen, you can post your answers by carefully reading the questions, because it is a situation based question. You have to read it twice or thrice to answer it. And your answers are option E and option D. Okay, let's see the answer. It is option E. This study can determine if ABC is horizontally transferred. So this is the not likely an outcome from the experiment. And why it is? Because the conserved gene cannot provide an adequate information about the taxonomic identity. Furthermore, they are saying like they will be running the two parallel experiment targeting separate gene. One is the 16S rRNA, other one is the ABC one. So it doesn't allow us to confirm if the read of these two genes originate from the same cells. So that is why this study shall only provide the information about the total taxonomic composition and the abundance of BC1, not the horizontal transfer. So this is why the option E is false. That is, it is not a likely outcome. So what about the other options? If you see the other options, the 16S rRNA, as we know, highly conserved regions across the bacteria allows for broader taxonomic classification, right? So that's why we will be getting higher number of OTU when compared to the ABC1 gene analysis, where a target resistance conferring bacteria. The ABC corresponds to antibiotic resistance in bacteria. So there will be higher OTU for taxonomic classification than the ABC1. Whereas in the option B, the 16S rRNA helps to identify and classify the bacterial community based on their taxonomic relationship. So that's why it is one of the outcome, likely outcome. And if you see the option C, by targeting the ABC1 gene, the analysis directly measures the prevalence of bacteria harboring the antibiotic resistant gene markers. So this tells us the abundance of such gene in the microbial community. Whereas in option D, yes, this is true because it cannot pinpoint an individual microbial species that are carrying the ABC1 gene without having the further analysis using the advanced techniques. That is why the option E, this study can determine if ABC gene is horizontally transferred or not. So we will move on to the next question. So which one of the following options present the correct order of steps that are involved in the whole genome analysis? So among these statement, you have to find which is the correct step to follow the whole genome analysis. Option A, first is DNA sequencing, followed by DNA fragmenting, assembly, and finally phylogenetic binning. Option B, DNA sequencing, assembly, phylogenetic binning, and DNA fragmentation. Option C, DNA fragmentation, DNA sequencing, assembly, and phylogenetic binning. Whereas in option D, DNA fragmentation, DNA sequencing, phylogenetic binning, and assembly. Whereas in option E, assembly, phylogenetic binning, DNA fragmentation, and DNA sequencing. So among these sequence, how whole genome metagenome whole metagenome analysis is taking place, and you have to find the correct order of step. And you can post your answers now. Find the correct order of steps that are involved in whole genome analysis. And your answers are A, option C. Any other answer? Any other answer? Option C. Okay. So now we will see the answer. Yes, it is correct. Option C. Now we will see the detailed solution for that. Because in whole metagenome analysis, first what will they do is, first they will try to extract the DNA and they will do first DNA fragments. So they break down the larger DNA molecules into smaller fragments which will be efficiently sequenced. So first is breaking the larger fragment into smaller fragment, then DNA sequencing. So this small fragments will be sequenced through NGS to determine the nucleotide sequence of each fragment. Then they will do the assembly. Once it has been sequenced, these fragments then computationally they will assemble. Then they reconstruct the original genome by joining this fragmented DNA sequence. Then at last they will be going for phylogenetic binning. What is that means? So the assembled genomic fragments they will be grouped based on their sequence similarity. It is like OTU, operational taxonomic unit. So finally these allows us to identify the individual microbial species within the community. So this is how the whole metagenome analysis takes place. First, what will you do? First, you will be breaking down the larger fragments into smaller fragments. Once you got these smaller fragments, you will be sequencing them through NGS in order to find the length of the each fragment. Once you find the length of the each fragments, then you try to assemble them and reconstruct the original genome. Then once you know the sequence length and reconstructed, then you can group these genomes based on their sequence similarity, which helps us to identify the microbial species. Yes, you are correct. Option C is the correct answer. Now we will see the next question, which is whole metagenome based analysis provide information about all organism, all microorganism, all prokaryotes, all bacteria, all bacterial chromosome. So whole metagenome based analysis provides information about what? Whether it give information about all the organism or only microorganism or only prokaryotes or only bacteria or only bacterial chromosome. You can post your answers and your answers are option B, option A. Okay. Now we will see the answer. Option A is the correct answer. Why? First, before that, you need to understand the term metagenomics. What it is, it refers to the select study of all the genetic material from all the organisms. So if you are taking an environmental sample, it will contain all the organism. It may include some plant samples also. So that's why here metagenomic, it will study all the genetic material from all the organisms that are present in the specific environment. So this means it not only includes the bacteria, it also includes archaea, eukaryotes, viruses, even other microorganisms like fungi. So under ideal condition, what happens? The whole metagenome contains genetic information of all the organisms whose DNA can be recovered from the sample. That is why the whole metagenome applies to all the microorganisms. Yes, option B is can also apply, but metagenomic primarily focus on the microbial communities. It also includes the genetic material from the other organisms too. So, like eukaryotes, viruses, etc. That's why the option A is more accurate than the option B. Whereas in option C, it not only applies for prokaryotes, as we have said, it also extends to eukaryotes, fungi. So, it covers the wide range of microorganisms. And option D, it's obviously not true because not only bacteria, it also contains all the microorganisms. So bacteria is small portion of prokaryotes. So that is why option D is also false. And option E, so what does it tells? All bacteria chromosome, it analyzes the collective genetic material from the entire microbial community, which is the metagenomic approach, not just the chromosome within the bacteria. So that's why all B, C, D, E is false, whereas A is the correct answer. Then the next question is bioremediation was first applied to treat option A, oil contaminated water, option B, plastic pollution, option C, municipal wastewater, option D, industrial wastewater, option E, heavy metal contamination. So, among these, bioremediation was first applied to treat which one of the contaminants? You can post your answers. Your answers are bioremediation was first applied to treat option C. Okay. Any other answer? Option B. Option C. Okay. Option. Any other answer? Municipal wastewater. Okay. Any other answer? You can go through the question. Bioremediation was first applied to treat. Okay. So now let's see the answer. Yes, it is correct. Option C, municipal wastewater. So earlier in our lecture, we have studied the evolution of bioremediation, right? Where what it happens, it started in the early 1900, where the first bioremediation was applied to the municipal wastewater. Then only they applied to the industrial wastewater. Then they have used for oil contaminated. Then only they slowly they have used hybrid approaches. So if you see, the first application of bioremediation is that municipal wastewater treatment. So that is why the municipal wastewater treatment is first applied. Then industrial wastewater, then oil contaminated, then it was applied to the heavy metal contamination, followed by the plastic pollution. So yes, it is correct. Municipal wastewater. Then the next question, which of the following statement about bioremediation is true? I will repeat the question. Which of the following statement about bioremediation is true? Option A, bioremediation always requires human intervention. Option B, bioremediation cannot completely eliminate pollutant. Option C, to microorganism, pollutant are always toxin. Option D, bioremediation may also be performed by insects and plants. Option E, bioremediation always involves the total elimination of toxicity. So which of the following statement about the bioremediation is true? It's option A, bioremediation always requires human intervention. Option B, bioremediation cannot completely eliminate the pollutant. Option C, to microorganism, pollutant are always toxins. Option D, bioremediation may also be performed by insects and plants. Option E, bioremediation always involves total elimination of toxicity. You can post your answers and your answers are answers are okay. Option D. Any other answer? Any other answer? Option D. Any other answer? Option A. Okay. So now we will see the answer. Correct answer is option D. Bioremediation may also be performed by the insects and plants. And if you see the detailed solution, first you need to understand what is bioremediation. As in the earlier, uh, we discussed on bioremediation, where it utilizes microorganisms like bacteria and fungi to degrade the pollutant, and it also extends to plants and insects. Also, we have seen the plants, that is phytoremediation, and insects also are being used in order to carry out this bioremediation process. For example, if you see the plants, what happens here? As we seen in the phytoremediation concept, it can absorb the pollutant from the soil through their roots. So they accumulate this pollutant in their tissue. Then it can be harvested and disposed safely. So this kind of pollution, this kind of remediation, we call it as phytoremediation, and it does by many plants. Whereas in case of insects, what it happens, it contribute by breaking down this certain pollutant by secreting certain enzymes in their digestive system or by exposing the pollutant to the environment by breaking down the organic matter. That's why the bioremediation is also performed not only by the microorganism, it also extends to the insects and plants. Now we will see why other options are not as accurate as D, because if you see the bioremediation, yes, the human intervention often needed for the design and implement of bioremediation. But there are, as we have discussed, the in situ remediation. There are some natural bioremediation process that also occurs without any human intervention. So that's why, but in the statement they have given bioremediation always requires. It need not be always, some of the natural process is there for bioremediation. That is why option A is false when compared to option D. And option B, bioremediation cannot completely eliminate pollutant because it can reduce the contaminants to the safety level, often rendering the site for reusability. That is why option B is also false. If you take option C, some microorganism can use these pollutant as a food source for their carbon and energy source. So they need not to be toxic for all the microorganism. And option E, bioremediation, what happens? It aims to reduce or remove the pollutant, but resulting byproduct can also be toxic, but it will try to minimize the toxic to the safety level. So this is not always true. They do not always eliminate the toxicity. Either they reduce, contain, or try to transform the pollutant. That's why option D is more accurate than all other options. So the last MCQ for the today's session is a pond is contaminated with industrial wastewater that is containing dyes. To treat this, the dye degrading bacteria is added to the pond water. So this is an example of option A, in situ intrinsic process. Option B, ex situ intrinsic process. Option C, in situ engineered process. Option D, ex situ engineered process. So I will explain the question. So here there is a pond where it is contaminated by the industrial wastewater. That too particularly dyes. So we will take an example of in textile industry where dyes are most oftenly used, right? So due to the textile effluent which containing dyes, it is the nearby pond get affected. So what happens here? Scientists they are trying to use a dye degrading bacteria into the pollute into the pond to treat this water. So this situation comes under which category? Whether it is an in situ intrinsic process or ex situ intrinsic process or in situ engineered process or ex situ engineered process. And your answers are option C. Okay. Any other answer? Any other answer? Option C. Okay. Any other answer? Option A. Okay. In situ intrinsic, in situ engineer. Okay. So now we will see the answer. Correct answer is in situ engineered process. So before moving on to that, we need to understand the different terms. First, in situ, it is an on-site or in place. Whereas ex situ means we will be taking the pollutant out and will be degrading them. Whereas engineer is we will be using some genetically modified or we will be isolating a particular microorganism for removal of contaminants. So why it is in situ engineer? As we are treating the pollutant on the site itself, that is the pond itself, by not removing the contaminant out from it. So it comes under the in situ. And why it is engineered? While maybe in the pond itself, there will be numerous naturally occurring bacteria. So if you are using naturally occurring bacteria, then it would be intrinsic process. But here, the scientists are deliberately adding and degrading bacteria to the pond water. So what happens? Here, their addition to the pond is deliberately euchromatic intervention to enhance the degradation process. So it comes under engineered process as opposed to the purely natural degradation process. If the question contained naturally occurring bacteria, then your answer would be in situ intrinsic. But here, scientists are aiming for particularly degrading bacteria. That's why it is called as engineered process. Since the biodegradation is carried out in the original contaminated habitat, which is pond, it is in situ. So additionally, we are trying to add a degrading bacteria too. That's why it is called an engineered process. So this is why the in situ engineered process is correct for this situation. So this is all for the today's class, and these are all the sample MCQs, and you have answered it well. So I hope you have also completed all the assignments on time and try to attend the lectures, all the lectures. It would be very beneficial for you because lot of sample MCQs are discussed during the live interaction session. And tell your friends also not to miss out these sessions. And if you have still any queries, you can always email me in the ajitveru284@gmail.com. And if you have missed out my earlier videos, you can go to my YouTube channel. You can visit these videos as these are all recorded sessions. And if you want to access these videos, you can go to the NPTEL Environmental Biotechnology course where you will have a recording sheet where I will be posting all these PPTs as well as the recording session videos. So you can access at any time, at any number of times. So this is all for the today's class where we have discussed the importance of bioremediation in environmental biotechnology. And as we proceed further, we will be dealing with the application part in the upcoming weeks, week 10, 11, and 12. So stay tuned. We will be discussing more real-time scenarios as well. And also we have discussed the sample MCQs. So this will be pattern of my teaching where I will be focusing on first the lecture recap followed by the MCQs. So that's all from my side. Have a great day. And if you have any queries, you can unmute your mic and you can ask. Or if there is no query, we can wind up this session. And if you still have any queries, you can post your queries in the discussion forum too, where our team will try to address all the queries on time. So, any query? Okay. Thank you. Thank you, Ashwini, and thank you, Nakati. Sorry for the mispronunciation. Thank you all. Thank you for attending these lectures and try to attend the lectures in the future also. Thank you. You can leave this meeting now.